Hello students, do you know what your blood group is? Yeah, obviously you know, but have you ever found out by yourself? So today we are going to do a fun biology experiment which usually every pathology which you see are doing every day. Now in this experiment we are going to see what my blood group is. So I'll give blood here and I'll check the blood group. Now what is blood grouping? So to know that we have to go back to 1901. So when a Austrian scientist named Karl Landsteiner, he was a physician and he tried mixing sample of different animals or different humans to find out that when few samples are mixed there is no clumping of blood, but when some samples, suppose my sample with another person, say X, someone's blood is mixed with my blood, sometimes we'll see clumping and sometimes we'll not see clumping. Now it 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 happens that on the surface of RBC, that is red blood cell, you all know maybe from class 7, class 8 standard. So the membrane of a cell, the cell has membrane, we all know it most probably. So cell has a membrane made up of protein, lipid and carbohydrate. Now the membrane has some sugar. Now those sugar molecule present on the membrane of RBC can react with some protein present in plasma. Now we have to recall again, blood consists of cells and a fluid called plasma. Now the cells have RBC, WBC and platelets. Now this RBC we are talking about right now in this blood grouping experiment. RBC membrane has some sugar. Sugar can react with some protein present in the plasma. Now the plasma of blood contains some protein and these proteins are called antibody. Now antibody can react with those sugar present on the membrane. So antibody and those sugars are now referred to as antigen. Now antigen antibody reaction is the basis of immunology. We know that in 2025 the Nobel Prize went to Shimon Sakaguchi and he is also an immunologist. So immunology is picking up the pace right now. And 100 years back, the experiment to find out what is the blood group is also a basis of immune system. Now let's not bore you with all this different theory. You can find theory even while searching chat GPTs or maybe perplexity. But we are doing experiment and find out how blood groups are determined and how what is my blood group. So I'm excited, although I know it. Okay, so I have a pricking device and I have my finger here. Now I'll be pricking my finger. Let's see how much blood do I have. It was really painful. Okay, so let's take some blood. So actually I have kept normal saline, which is 0.9% sodium chloride, you all know, NS. And this is my first drop of blood. It goes here. And this is my fourth drop. It was really tough. Okay. Now I'll explain why I have taken fourth drop. But first, let's do the experiment first, because if I delay, the blood might clot spontaneously. So now I have this antibody, and this is NTA. I'll explain what is NTA. Now here I'm taking a drop of NTA. Now this is antibody of B. This is anti-RH. This is antibody of RH. You don't need to worry about the terms. I'll just see. Now I've kept 2 extra drop of blood to find out whether they are clumping automatically, like spontaneously or not. I've put anti-A, means antibody of A. I'm mixing it to find out. Yeah, you can see my blood is totally clumped. Can you see clumping? Now, the same way, if I mix this one where I have taken B, see, there is no clumping, there's clear cut difference. The blood drop where I have put this anti-RH, there I can see again there is no clumping. We can see here that the blood drop where I have mixed this antibody of A, that drop has lot of clump. And the drops which I have taken and didn't mix any kind of antibody, the drop here also, there is no clumping of RBCs. This means my blood group is A. So for there is A type of antigen in my RBC, there is no B type of antigen on my RBC, and there is no clumping where I have put this anti-RH. This turns out that my blood group is A and RH negative. And so the drops where I have not put any kind of antibodies, so any antibody solution, those drops didn't clot. Now what happened? What is the principle? Here, this is the blood cell, and blood cell has this A sugar, A antigen, and this is the antibody it is mixing with. It binds to this blood cell, and the B antibody couldn't bind, and this is where we can see that this is A type of blood group. But there is another antigen which is absent, so that turns out that it is absent in my blood, but it is present in almost 85% population, and that is Rh antigen. So when Rh antibody was given, this Rh antigen binds to that blood and clumps the blood. So now if you have B positive, that means your blood has this B antigen and your blood has this Rh antigen. These two antigen interacts with the antibody and gives us the result. So if you have learned something, we are coming up with a series of experiments where we will find out how plants communicate with each other, how human nerves are interacting, how synaptic junctions are formed with some kind of experiments. This will turn out to be an exciting series of experiments. It will be fun. And thanks so much for watching. This is Dr. Saikat Kar, neuroscientist, Human Brain Project, but most importantly, your mentor in learning few great experiments.